Okay, everyone, we're just going to quickly go through the first set of these notes. You've seen them before, and so if you want to just scroll along in just a review as we do this, that'd be great. Okay, so inheritance. So far, the ones that we still need to cover, I think, is the sex chromosomes, 2.5. We need to look at Punnett squares. We need to look at chromosome mapping. Those would be the so 2.5, 2.3s, and 2.3s4. Those would be areas that we need to look at, spend a little bit of time. And also, I think we need to spend a little bit more time on 2.4. Okay. So we start off by talking about the fact that all humans have been doing this for a very long period of time. They just haven't formalized it. We've known about it, say, for example, with dogs, and I've talked to you about that. Selective breeding to get all the different variations. The first person who did this formally, Gregor Mendel, used peas for reasons that are shown here, that they've got easy characteristics. And remember that these are genes. And the two different forms of the gene that are easy to work with are called alleles. Don't get those two terms confused. They're very key. And we assign the dominant is a capital and the recessive is a lower letter for the dominant allele. So yellow would be big Y, and green would be little y. Okay? They can cross and self-pollinate, and they have short generation times. Cross-pollinating means this. There's my cross-pollinating, and self-pollinating means this. Okay? When it's crossing by itself. Okay, we did this cross here where we took purple true breeding combined with recessive of that gene, and we get hybrids. And when we self-fertilize these, we showed the Punnett square where we separated the alleles into the two different types of gametes, dominant recessive, the two types of gametes, female on the top, male on the bottom, and we got the allele pairing combinations like so, and lo and behold, the recessive trait shows up. And so we know that homozygous recessive is the only genotype where we know, or the only phenotype rather, where we know the actual genotype. Here's the phenotype, what we can see, and here is the genotype, the allele pairings for that phenotype. And we know what that is. Here we don't know whether it's going to be dominant or recessive. We just know the probabilities. That's all based on our Punnett square. That's going to show that it could be 25% chance pure breeding, 50% chance that they are heterozygous, and then 25% chance for homozygous recessive. So we've covered through that a lot of terminology that we've gone through. Allele on homologous chromosomes. We've talked about the law of dominance. We talked about the law of segregation and meiosis, and independent assortment in meiosis. We spent quite a bit of time on those already. So just read through those notes one more time. And then we looked at the nomenclature: homozygous dominant, heterozygous, homozygous recessive, genotype, and phenotype. And we've already interpreted Mendel's results. We've talked about how to properly do a Punnett square and what it actually means. We did a corn lab that was a one-trait cross with color, where it was 3 purple to 1 yellow. We talked about a test cross that I don't know if this puppy is pure breeding or heterozygote. So we cross it with a known genotype, homozygous recessive. Okay, and if the recessive allele shows up again in the F1s, that tells me that this dog is actually heterozygous, not pure breeding. But if the dog was pure breeding and we cross that with a recessive, homozygous recessive, that means that all the F1s must be dominant, in this case, black. That tells me my dog here is purebred. A very racist connotation to it, pure breeding, pure bloodline. We know in actual dogs that's a huge problem, leads to all kinds of genetic problems with inbreeding. So pure breeding is not the best. Get a mongrel. Okay, so there's that. Dihybrid crosses, two trait crosses. Remember, the key thing here is that these are on different chromosomes. Different chromosomes. T is on, height is on one, color is on the other. So that these chromosomes here, it might be on chromosome number 5, this is on chromosome number 1, they are on separate chromosomes, so that's why we write both allele in the gametes. Okay, and we said with these, when we combine two heterozygotes that are dominant and recessive, okay, two heterozygotes. It's the first heterozygote, there's the second heterozygote. When we combine them, we will get a chart with 16 boxes in, and we get this 9 to 3 to 3 to 1 ratio. And we practice that again, where with these wrinkled ones, yellow and sweet, okay, that would be yellow, and we called it wrinkled, so that would be little w, or smooth, so that would be wrinkled, so little s's, whatever you wanted to denote it, that would be, we would get 9 to 3 to 3, and approximately 1 there. Okay, there they are. Incomplete dominance, we said, was a blending, so we get a new phenotype forming. Okay, and you can get that in, say, flowers, where this really should be big P, big W, because we're getting a blending of these traits. R's, so we get RW. Okay, one is not dominant to the other, they're blended. So I don't really like how one of the books has shown it this way. I prefer to have big R, big W, big R, big W, and these are two W's. They're not recessive. Okay, notice that the ratio here is a 1 to 2 to 1 ratio. Yeah, okay, we saw this in sickle cell anemia, sickling trait. Okay, actually confers resistance to malaria. These folks have a short lifespan. These folks have normal hemoglobin. And then codominance. We don't get a pink cow, we get red and white. So both alleles are fully expressed. There's no blending. And we would get this here where these folks are not pink, unlike the flowers, they are roan. Both get expressed. A more common one, of course, is ABO blood grouping. And this gets to something we've only spoken to a little bit, is that we have a hierarchy with 3 alleles here. A is codominant to B, and they are both dominant to blood type O, which is designated at little I. There we go, blood type O right there. Okay, be prepared to write several exam questions where a person, say blood type A, marries a person that's blood type B, and yes, they can produce a child that's blood type O because they both carry the recessive allele. Okay, so that is the review, and I'm going to leave that as the first video. You can make sure that you've got all those questions done. The next video, we're going to go on to Mendel's laws of sex-linked, and we'll look at chromosome mapping on the next video.